right, well, welcome back everyone to the MSU Science Festival Going Digital live presentations. And I'm Roxanne Truin, with the coordinator of the festival, and I'm with Michelle Russell, who is the graduate coordinator, academic advisor, molecular diagnostic co-instructor with the MSU Biomedical Laboratory Diagnostics Program, one that I'm a proud graduate of many moons ago. And she's going to walk us through how to extract DNA from a strawberry in the comfort of your own home. And Michelle, if you could give the list they need to do their demonstration, and then I'll ask you to tell us a little bit more about how you got interested in what you're doing.
Sure, thank you, Roxanne. So the materials that we need today for your DNA extraction are actually really simple. When we first practiced this, I don't even think we had to go to the grocery store, which was really nice. We had a lot of these at home already. We're going to start with 2 strawberries. We're going to need a Ziploc plastic baggie. We're going to need some dish detergent and salt. We're going to need a couple measuring cups. We're going to need to measure different amounts of teaspoons today. So we don't have a lot of measuring cups around the house, but we do have kids, and so we have these little medicine cups. So we have lots of those, and I've marked teaspoon where the teaspoon line is. And if all you have are cups with milliliters on it, about 5 milliliters is about equal to 1 teaspoon, and that'll work perfectly for today. We also have a couple of clear plastic cups. One of them has a 1/2 cup of water that I've prepared. We're also going to need a spoon, a coffee filter, and some cold isopropanol. As you can see, I've had it in ice water, but before that it was in the freezer. If you just have room temp isopropanol, that should work pretty well also. And what else? We need a stick, either a coffee stirrer, or I have a skewer, for barbecuing, and I just broke it in half. I took the pointy part off. So those are about the things we need for today.
Awesome. It looks like we'll have a great experiment coming up. So Michelle, can you tell us what is a clinical laboratory scientist?
So a clinical laboratory scientist is a person who works in a laboratory, often running tests on human samples to help detect certain disease states. We do a lot of testing in medical microbiology. We do testing in blood banking, clinical chemistry, hematology, and those are all very specific areas. They help doctors make clinical decisions about how to treat patients with disease. So we do a lot of behind the scenes testing, but we play a very important role in clinical decision making.
Awesome. Before we get further, we did have a question pop up that says, will it work with other fruits like blueberries or frozen fruit?
So if you have frozen strawberries, it should work pretty well. The reason why we're using strawberries today is because in the cells of strawberries, they actually have 8 times the normal copies of DNA compared to human cells. So if you have strawberries, it would probably be best. They just are jam-packed full of DNA. Can try for blueberries, but if you have strawberries around, that's probably ideal.
Okay, and they could be a real scientist and experiment and see if it really works with a blueberry, which scientists, that's one of the things they do is they test out different methods. So this could be a different method for you. Mm-hmm. Awesome. All right, so you were mentioning that clinical laboratory scientists do biomedical medical testing. Are they involved with the COVID-19 pandemic at all right now?
Absolutely. In fact, it's the medical laboratory scientists that are doing the COVID-19 testing on all those patient samples. So they're the people that are doing the scientific analysis and have such a huge impact on healthcare.
Great. And how did you get started? How did you want to be a laboratory scientist?
So I was always interested in science. It was my favorite subject in high school. And when I went to college, I knew I wanted to go into like a science-related healthcare profession. And I tried a couple different things, but I ended up finding medical technology. That's what it was called at the time. And I'm so thankful I found it because it's that perfect mix of science, problem solving, critical skills, but it also has a direct correlation to pathology or the disease processes going on in the human body. And I just thought it was so interesting. And lab testing really does have such an impact on patient care. So it was a great fit for me.
Awesome. Awesome. And how long does it take to be a clinical laboratory scientist?
Well, most programs to be a certified medical lab scientist are a 4-year bachelor's program. That's the academic part, but then there's an internship requirement that most people do. That's usually about an extra 6 months to 10 months. So it's a bachelor's level type of program. And once you're done with your bachelor's program and your certified or your accredited internship, you're eligible to take the national certification exam. Most hospitals require a certified professional or a licensed professional.
Awesome. All right. Well, I will let you get started. And who's your helper?
This is my son, Ryan. He's helping me today. We practiced this before, and I thought he'd be a good helper, and he was interested in it too. So Ryan's going to help us today, and we're going to be able to go through the steps one by one.
Awesome. Thank you. We're excited.
Okay, so now that we have all of our materials in front of us, we're going to start with the procedure, which is Ryan is going to take the greens off of the strawberry. So go ahead, Ryan, why don't you do that? We're just going to put it into a little plastic container. Otherwise, you could just walk it to the garbage, set it aside. There you go, Ryan. Okay. So as he's taking the greens off, that'll allow us to really focus on the strawberry cells. And then Ryan's going to put the strawberries into the plastic baggie and zip it up again. And then he's going to crush it for about 2 minutes. That's really going to help break up the cells, break up the strawberries so that when we add our DNA extraction mixture, it'll really be able to access those cells even further. So Ryan, let's have you come over here a little bit. There you go, buddy. Ryan's just crushing it. This is like the fun part for the kids, right? It's fun for me too. He's just using his fist. And as he's breaking up the strawberries, it'll allow us greater access to those cells so that when we add the lysis reagent, they can really lyse those cells really well. You know what I'm going to do? I'm just going to open up this bag for a minute, and I'm going to just press out some of the extra air. That'll help you really get to all those strawberries. There you go, karate chop it. There you go. And for anyone watching at home, there's no perfect way to do this. This is why this is such a fun experiment. If we're in a laboratory extracting DNA, it's much more controlled. We typically extract DNA from blood or bone marrow, other types of samples. But today, strawberry DNA is a great way to do a safe experiment at home and still demonstrate the principles of DNA extraction. In the lab, we sometimes run DNA testing to do different typings on people or microorganisms. It helps inform physician decisions on how to treat infections, either in humans at various levels. All right, are they pretty crushed? Yeah. All right, so now we're going to leave that bag of strawberries right there, and we're going to make our DNA extraction mixture. So we already have our water. I already pre-measured our half cup of water. We'll set that aside. So Ryan, why don't you pour out 2 teaspoons of detergent, okay? Let's switch this around, that's easier. Can you measure up 2 teaspoons up to that line right there? Yep, see where I mark that? Okay. All right. And then measure out 1 teaspoon of salt there, okay? Yep, yep. Perfect, all right. So what you can do now is dump your detergent and your salt into the water, okay? You can use your spoon maybe to scoop out any extra detergent. There you go. Okay, that's okay. And then dump your salt in also there, bud. Okay, now you can start mixing it, okay? Give it a good stir. let's see if we can dissolve that salt. So as we make our DNA extraction mixture, we can use this to kind of pull out the DNA from the cells. The detergent and the high salt concentration actually help to lyse the cells of the strawberry, and that lets us access a large amount of DNA that are in the strawberries. There you go.
So when you say lyse the cells, what do you mean by that?
So that means break apart the cells, literally blow them up, right? Because the DNA is in almost every strawberry cell. So you need to blow up the cells, break up their membranes in order to release the DNA. That's okay, don't worry about that. Give it a couple more stirs and then we'll go ahead and get that going. So you can probably see from Ryan's mixture, it's kind of cloudy. It's a little sudsy, and that's kind of what you're looking for. The salt might not totally dissolve, but the mixture is looking pretty good right now. All right, Ryan, so what we're gonna do, now we're gonna add 2 tablespoons of the DNA extraction liquid to the bag with the crushed strawberries. So let's see here. So you see the 2 tablespoon mark? I'm gonna just pour this for you. Hold up the bag of strawberries there, bud. Can you hold it up a little higher so everyone can see? I'll do this part while you hold it. We're gonna just add 2 tablespoons here, or 2 teaspoons, sorry, 2 teaspoons, better get it right. I'm gonna dump it in. I'm gonna close the bag and try and eliminate some of the air because we don't want extra bubbles in this bag while we're crushing it. And so what Ryan's gonna do is he's gonna mix the DNA extraction liquid with the strawberries for about a minute. That'll give time for the extraction mixture to really lyse those strawberry cells, release that DNA into the solution. And then the next step is we're gonna strain the strawberry liquid and then add some isopropanol so that we can precipitate out the DNA.
Awesome. I had a question that says, I just turned on and don't know exactly what they are doing. Could you occasionally mention the purpose of the procedure? So if you want to just give a recap.
Sure. So we're doing a strawberry DNA extraction here just to demonstrate how easy it is to do the DNA extraction. But in the medical laboratory, we extract DNA all the time for a lot of different reasons. In light of current events, there's a lot of medical laboratory scientists doing DNA extractions from patient nasopharyngeal samples in order to try and extract that COVID-19. I think it's RNA actually. So that we can determine if a patient is positive for COVID-19. In a lot of different applications, we're extracting DNA so that we can do microorganism detection and identification in human samples for infectious disease testing. We also do DNA extractions and tissue typing and organ transplantation so that we can HLA type different donors and recipients to make sure that there's a good match for organ donation. And in molecular diagnostics, there's a lot of DNA typing going on in tumors so that doctors can better treat types of cancer that patients are going through.
Great, thank you.
So Ryan, I think we're ready to strain out the strawberry extraction liquid. So now we're gonna take our other plastic cup and a coffee filter, and we're gonna dump in that DNA extraction liquid and strain out the liquid part from the pulpy part. There you go, Ryan. This can get a little messy here, and it doesn't have to be perfect. When we did it the first time, when we practiced, it was not perfect. We had to dump it a couple times, but that's the nice part about science, and especially like DNA extractions, you can usually fix issues that come up. You might be limited by your sample in a clinical lab. Certainly here we have lots of strawberries. There you go, honey. That's okay, just set that down. And Ryan, do you remember how we twisted it? Yeah. Okay, we're gonna kind of keep the top part together and we're going to let that strain out. In a laboratory, you can often fix problems if they come up. And that's a great part about being a medical lab scientist is that you're always problem solving. You're always using your critical thinking skills. You do need to follow the procedure in the lab, but when problems come up, you really are challenged every day and it's an interesting profession to be in. All right, Ryan, you're doing great. As you can see, Ryan's getting a lot of strawberry extract. And he'll just kind of keep gently pushing that coffee filter. I think it really helped that we practiced this a week ago. We know what we're doing now. And that actually speaks true for a lot of laboratory procedures. The more you practice, the better you get at it, the more efficient you become at it. You really become kind of a master of your craft. Before I came to education, I was in the medical lab probably for about 12 or 13 years doing different types of things. And it was always fun to have a new procedure in front of me. It was a new challenge. And analyzing the information that we got from the test result was always something that kept me interested in what I did. There you go, Ryan.
So you're not just doing numbers, you really have to think about the patient and everything that's involved. And if the result seems a little odd, you do some backtracking to try and determine what's going on.
Why don't you go put this in your cup here? Go rinse your hands off real quick. Yeah, absolutely. You're always kind of looking at the overall clinical picture, looking at test results from different areas of the lab to make sure your result makes sense. And that's why it's so important when you go through your medical lab science program that you're doing, you're taking coursework in microbiology and hematology, clinical chemistry, and blood banking and other disciplines, because those all inform kind of the whole process going on in the human body. All right, so now we have our strawberry extraction liquid. This is excellent. We're gonna take our cold isopropanol now. You sure can. And what we're gonna do is we're gonna add an equal volume of isopropanol to what we have in volume of the strawberry extract. So if we have about this much of our strawberry extraction, we're gonna go just about half, a little bit more up. It really is about eyeballing it. You know what, I'll draw a line on here. So here's our strawberry extraction. We're gonna go about up to here for the isopropanol. You see that, Ryan? Yeah. Okay, go ahead. Squeeze the isopropanol in there, buddy. It's okay. So in a medical lab, we use some very similar reagents. We actually do use isopropanol. I'm going to just start swirling this gently because that's going to allow it to mix and start to precipitate out that DNA. But in a clinical lab, we're using isopropanol. We also use ethanol. We often use proprietary reagents that we buy, but you could make some too if you needed to in a lab. Sometimes it's a little bit more cost effective. But it's really just about the chemistry of each step. You want each step to go well, and you want each step to do its job so that the next step will work. We're starting to see a little bit, let me see here if you can see, a little bit of that white solution at top. That's our DNA. So we're gonna swirl a little bit more, and then I'm gonna have Ryan pick it up with a wooden stick. You'll see that we just got globs and globs of DNA from our strawberry extraction. Whichever side you want, buddy. Oops, I'm gonna swirl a little bit more. Okay, I'm gonna help, and then I'll pass it over to you, Rye, okay? So what you're gonna do is you're gonna take your strawberry extraction liquid, and you're gonna swirl that big white blob around your stick. Okay, Rye, take it over from here. So Ryan is holding up, I'm gonna hold the cup under there. Ryan is holding up our big glob of DNA. That is a huge amount of DNA. That's why strawberries work so well, because they have so many copies of DNA in every cell. But I hope if you're doing this at home, you were able to follow along. And if you couldn't follow along today, there's instructions, I believe, posted to this presentation, and they're really easy to follow.
That's correct. They will be, and we can also post them on our website somewhere. We'll find a spot for it.
Wonderful.
Awesome. And so if you were in the clinical lab at this point, would you do something with that DNA then?
So oftentimes we do testing on the DNA. Sometimes we need to purify the DNA a little bit more so that it's appropriate for testing. But at this part, once you get your extracted DNA, you can then move downstream with your testing. So many different things you can do with testing DNA. You can test for very small, for very targeted genes that might indicate disease, or you can test very large genes that have lots of mutations. So for example, sometimes when we're testing in cancer diagnostics, we're just testing for a mutation, maybe in something called an EGFR gene or a KRAS gene, and the results of that testing might indicate a certain therapy or not. Sometimes when we're testing for inherited mutations, there's really large genes that we're testing. For example, the cystic fibrosis disease, is caused by mutations in the cystic fibrosis transmembrane gene, I believe. Of course, I'm drawing a blank when I'm talking about it live, but it's a CFTR gene. There's up to about 600 mutations in that gene that can lead to a patient having cystic fibrosis. So
Wow.
that's a really large gene.
That is a large gene. I did have a question. I think they came in late. Can you use other kinds of fruit? So if you want to just kind of reiterate why strawberries work so well. And
yeah, so if you have strawberries on hand, those are really best to use in this type of situation because they have lots of copies of DNA in their cells. I think they have about 8 copies of the same DNA in their cell. It makes them a really robust cell to extract that DNA from. You know, humans only have 1 chromosome from mom and 1 chromosome from dad in each cell. We're able to extract DNA really well from blood or bone marrow in the clinical labs. We also use other samples like tissues or even like other like urine samples or wound cultures, that kind of thing. But when you're doing the DNA extraction in your kitchen, strawberries are ideal. You could try blueberries, you could try raspberries. You might want to look up and see how much DNA they have if you want to Google it.
Fruits have DNA.
Yeah, well, most cells have DNA in them. It just depends how much.
right, so you could create your own experiment and see which fruit has the most DNA, and that could be a whole different set. Maybe do 5 different fruits or and see which one has the most DNA. So another idea to, when you're staying home and staying safe, to do experiments out in the kitchen.
Mm-hmm.
Mix all the DNAs together.
You mix all, yeah, you can mix all the DNAs together too. That would be interesting.
We have about 8 minutes left, and if anybody has any questions for Michelle about maybe a career path or any more information they'd like about the clinical laboratory scientist pathway and what you would do once you graduate. As I mentioned, I was a former clinical lab scientist, and now I organize the Science Festival, so it can lead you in a lot of different directions.
Mm-hmm. Yeah, in fact, I'm on the same boat as you, Roxanne. We were talking before we started today. Not every career path that starts in the clinical lab, just because you start there doesn't mean that you might work there in that capacity forever. You can go on to pharmaceutical or in vitro diagnostic sales. We have students that go on to work for contract research organizations. So they help run clinical studies that bring kind of new drug testing through clinical trials and FDA approval. That's a career I didn't even know about when I was going through my medical technology program. There are a lot of different areas that you can specialize in the clinical labs. So I mentioned earlier, you can go into hematology where you're evaluating human blood for different diseases, maybe like leukemias or lymphomas. You can go into specialty testing using flow cytometry. There are other methods also that you can use in the hematology lab, but you could also go into coagulation. So you can study bleeding disorders. Different.
All right.
Oh, go ahead.
I do have, Melissa wants to know, what stuff do you test? So I'm guessing she's asking, as a clinical scientist, which you just hit on, a lot of different things.
So in the clinical lab, we primarily test blood. Now, blood can be separated by different categories like plasma or serum. We can also test bone marrow, and we can test urines. We also are increasingly testing tissues in different capacities, especially in the molecular diagnostics lab. In the microbiology lab, we accept a variety of different samples. Sometimes we test samples from your nose or from your respiratory tract. We can also test stool samples, or even if you have a really bad cut that isn't healing, that's called like a wound sample, and we'll culture wound samples. But blood is the primary one in different phases.
All right, and another question, Alyssa's son wants to know what you like best about your job that you do now.
Oh, so right now I'm in education, and the part I love about that is that I get to help provide suggestions to students on their career choices in the medical laboratory. I get to help them choose their courses at Michigan State so that they're successful in their career or in their academic and career path. And I actually am really lucky to be doing some instruction in the molecular courses in our unit with another instructor, Dr. Morris and Dr. Gerlach, but I really love the science that I still get to do. I think I kind of have a nice variety of responsibilities that keeps me interested in every day and going to work and helping students, but also helping kind of create the pathways for professionals that are really needed in the field right now.
Terrific. We have another question. Diane's daughter wants to know if you can cross fruit DNA to make new fruits. I don't know if you can answer that, but
That might be more of a plant biology type question. And we do have a really great plant biology and biotechnology. We have those departments at MSU. That's not something we do a lot in medical laboratory science. We're more focused on understanding the testing that detects different biomarkers and chemicals and DNA in a patient sample.
and I know the presentation before yours, he actually was a plant molecular biologist, and he did talk a little bit about that. So if you want to look for that video, you can check that out. And he talks about peaches and plum trees and how they look different. So FYI. And we had one other question from Naringa. She wants to know, at what age can you start helping in the lab?
So usually we say probably want to have a high school education. You could probably do some, I take that back. So you could probably do some shadowing while you're in high school, maybe even middle school, but probably high school. You can go and observe what's going on in a clinical lab, but you could probably get a lab assistant job after you graduate from high school. And certainly after college, you could get a medical lab science position. There's also something called medical laboratory technicians, or MLTs, and that's also a little bit shorter pathway to working in the lab. But my background is more in the bachelor's level program.
Well, we just have about 2 minutes left. So I also wanted to do a little shout out for the rest of the day. We have another 4 hours of live presentations coming from the MSU Science Festival. You can find the lineup at sciencefestival.msu.edu. And Michelle, do you have a website that if people were interested in learning more about how to be a clinical laboratory scientist, they could go?
Absolutely. I can point you guys to the BLD, or Biomedical Laboratory Diagnostics program at Michigan State University. And there's a lot more information there about what our students do at the undergraduate and graduate level and the impact that we're having. Medical laboratory scientists are having a huge impact with the COVID-19 testing right now. And you'll see some of the other activities that our unit is doing, so that we're promoting the profession and trying to communicate to others what we do.
Awesome. And Melissa wanted to know if we can get DNA from cucumbers. Have you ever tried anything besides strawberries?
You know what? I haven't. I've been in the medical lab and I've always extracted DNA from human samples. And then this was our first foray into fruit DNA extraction. I bet if you tested the seeds, you might be able to find something.
Awesome. And then we had a comment that Ryan is quite a junior scientist. So shout out to Ryan.
Yeah, it's never too early to get your kids interested in science. And I was lucky that he wanted to help with this. So
awesome. Well, thank you, Michelle, for spending this half hour with us. We really appreciate it. It was a great demo, and it's something that families can now do at home when they're in their stay home, stay safe mode. Mm-hmm. And we want to remind everybody of that. And we thank them for watching. Any last comments?
No, just if you want more information, you can visit bld.msu.edu. But I can send you that link if that would be helpful. And I would, again, like to thank all of our medical laboratory scientists that are doing the work right now in the hospitals and labs.
And I would like to do that as well. When you hear about all the testing being done, those are the clinical laboratory scientists hard at work there, putting their lives on the line too, dealing with what, there are a lot of positives out there. So special shout out to them. So when they announce all the healthcare workers, be thinking of the lab people as well. Mm-hmm. All right, with that, our time is done. Thank you, Michelle and Ryan, for
Thank
your
you.
demo.
Bye-bye.
All right, bye-bye.
Thanks, Roxanne.
Thanks.